see how it relates to what is happening currently in our edge counties, our local counties, with regard to the badger cull. And so I will hand you over to Malcolm Bennet. Malcolm.
All right, well, thank you, David. Hello to everyone. And first of all, a big apology, because I was meant to come down and do this talk. I'm not quite sure when, but it was probably about a year ago now. And I managed to double book myself, and then this coronavirus thing got in the way. So I can't see any of you, which is awkward because there'll be no feedback whatsoever. So I'm going to share my screen and I'll just get on with it. And if you want to, as Harriet was saying, if you want to post some questions in there, do so, and then we'll try and answer as many as we can afterwards. If they're ones I can't answer and Ian can't answer, then we'll try and get back to you later with the answer to those questions. So I should be an expert at this by now because we're doing all our teaching online, but it'll probably go horribly wrong. Let me share my screen. And that's the one I think we want. Can you see a proper slideshow there? Say yes, somebody. Excellent. Yes. So if I do that, if I do that, it moves on, does it?
Yes.
Yes.
Excellent. Okay, so I will start on the first slide and just say, so I'm Malcolm Bennet. There are lots of other people involved in this, and I'll come to them later, but I do want to start off with a thank you to everybody, including lots of people in this audience, for collecting a lot of the material that we used in this study. I'll come on to why there have been so many delays in publishing it towards the end of the talk, but just to say up front that we have now finished all the statistical analyses. We're in the final stages of putting the paper together, and we want to submit that as well as soon as we can. And I think DEFRA are discussing at the moment publishing the report as soon as they can as well. So everything that's in here and plus a bit more will be out in the open pretty soon. Okay, so I thought I'd start off because I'm sure everybody here knows more about this than I do, but this is kind of the background as I see it. So bovine TB used to be a big problem in humans, not just in cows, and was almost eradicated by testing cattle and then culling them, and eradicated in people by pasteurizing the milk, because if you pasteurize the milk, the TB organism, Mycobacterium bovis, is killed, and then you can't have any infections in people. But back in the early 80s, maybe even the late 70s, more and more cases in cattle started to be seen. And some of these were traced back to badger involvement. So that epidemic in cattle has been gradually moving north since, and you've got some little maps down over here showing that, although only up to 2010. And in the endemic area down in the southwest where it started, then it has been suggested that badgers, and I put in brackets all wildlife or the environment, because it's difficult to distinguish these different things when people do mathematical models of what's going on. It's suggested that badgers might be responsible in those areas for infection of the first cow on the farm, but thereafter it's bovine to bovine transmission, which is the main route of spread. But what nobody has any idea of is what's happening at the edge of the cattle epidemic, which is the area outlined here with the black line. And I guess my feeling has always been, if you want to work out who's infecting whom and who's to blame, I'm going to put that in quotes, then you really want to look at where the epidemic is spreading. So is it more common in badgers and then it turns up in cattle? Is it more common in cattle, then it turns up in badgers, or what? And then the last point I put on there is that so we don't really know what's driving that spread in the cattle epidemic. And in the low risk areas, the areas in green and yellow on the lab, so that's low risk for cattle, because all of this is based on cattle, then it tends to be thought, because there are sporadic outbreaks in cattle, that's long distance transport of cattle from one farm to another. And usually that's backed up both by strain typing, the isolates of TB you get out of them, and by epidemiological tracing as well. Okay, so I've said that bit, and the other bit I wanted to highlight on here is this map here. I suspect, again, most of us have heard of spoligotypes, but spoligotypes is just a posh name for strains, and it's a way of telling apart different strains of bovine and human TB. And the reason that it's used a lot in bovine TB is it looks like different spoligotypes or strains are found in particular regions or areas of the country. And when you have a spreading epidemic in that area, it tends to be of that local or regional strain. And in case I forget to say it later on, that is quite interesting, because if this was an epidemic which all started down in the southwest, what we would expect is all of these to be exactly the same, or at least all be mixed together. And actually the fact that we have regional different colors for the different strains on here rather suggests to me that what we're looking at is regional epidemics rather than, and therefore a spreading north of different epidemics rather than one big epidemic spreading north, which is a bit kind of weird, trying to work out what might be going on there. Oh yeah, so the other thing I've put up on this slide over here is the area we were studying was, I said the edge, but we were only looking at the northern edge. There was another study of the southern edge, which has been done, and that was headed up by the University of Surrey. We were involved in that because we did a lot of the isolations of the mycobacteria from that study. But I'm not going to talk about the outputs of that study today. So our approach was, we thought, well, we should make use of roadkill badgers. Found dead is the other kind of politically correct term for that, because why would you want to hurt any badgers when you were doing this kind of work? And those badgers were collected by a network of stakeholders, because that gets people like you engaged. So thank you very much again to those of you who did get engaged, and some of you got very engaged and collected loads and loads of badgers. And I've met lots of lovely people who were quite happy to put a dustbin in their front garden for me to go and pick up, or my colleagues to pick up badgers from their gardens. So I suppose the next thing to say is just a little bit of background about roadkill badger surveys. It used to be that in the days of MAF, before DEFRA came along, MAF used to test badgers to see if they had TB. And they did that up until about 1990. So there's about 20 years worth of data there. And if you look at the maps of that overall, so the blue dots on this little map show you where they collected badgers, and the red dots show you where they were finding badgers with TB. And I guess my summary of that would be that they tended to find badgers with TB in areas where the cattle had TB. They did that again, and in Cheshire, which I'll come back to, in that period, it was 0.25% of the badgers had TB. They did a similar survey in 1991 to 1997, which I think is a completely pointless exercise, because there they only looked at the areas where the cattle had TB, and therefore they found it in the badgers where the cattle had TB. So it doesn't really tell you anything about spread or epidemiology. There has been, and there are ongoing studies of roadkill badgers in Wales, and I'll come back to that at the end because they've just published their latest data on that. And in 2014, we did, when I was at the University of Liverpool, we did a pilot study in Cheshire, which was not very popular with lots of people, but I still thought this was quite a useful thing to do. And we collected 94 badgers within the county, and I put that in quotes because some of them came from a bit across the border, and theoretically, there was no such thing as Cheshire anymore. It's two separate counties. But we found 22%, that's 20 out of 94 badgers with TB infection. So that's whatever that is, 100-fold increase since the 1972 to 1990 period. We have no idea what the gradient of change is over that time, but of course, by the time we did this in 2014, TB was really starting to take off in cattle. So all the badger isolates in Cheshire were spoligotype 25. So this strain, which is the same strain which was found in the cattle that were epidemically infected in Cheshire at the same time. The maps up the top there are risk maps for how likely were badgers and cattle in any area to have TB, and they overlap completely. So the conclusion from that we drew and that we published was what we're seeing in badgers, what we're seeing in cattle was probably part of the same epidemic, but we tried to make it very, very clear. Well, we did make it very, very clear, but I'm not sure that everybody wanted to read it, that didn't tell you anything about which way transmission was occurring. It simply tells you that this is probably the same epidemic that's going on. So I said I'd come back to the Welsh study. They published a study, I think in 2012, something like that. And then a month or so ago, I think in July of this year, they published a follow-up study. And this was the results from their first one, and this is the results from the second one. And the green dots on here, if you can see it, just about are where they got positive badgers. The blue dots are where the badgers were negative, and the black dots is the density of cattle in Wales. And I guess I would summarize this by saying that most of the positive badgers came from the area in South Wales, where there's a lot of TB in cattle, or along the border area with England, which borders onto the areas where cattle bovine TB is epidemic, so high risk area of cattle in England. So in some ways it's a bit similar to these other two studies in that you find TB in badgers where you find TB in cattle. You could turn that around if you wanted to, and so you find TB in cattle where you find TB in badgers, and that's kind of up to you how you interpret it. Right. So what did we do? So we had a roughly 1 year. Actually, it took a bit more than that project, and it was based at Nottingham, but involved Liverpool University, and it was core-funded by DEFRA. And I say core-funded because there was various other people who put money in, and of course, lots of people put a lot of time in, which ought to be counted as part of the funding. We collected roadkill badgers from the northern edge counties, and a lot of you will have seen this talk before, so I'm sorry about that, although I'm told you shouldn't apologise when you're giving talks, but anyhow. So those edge counties in the north were Cheshire, Derbyshire, Nottinghamshire, Leicestershire, Warwickshire and Northamptonshire. Collected by lots of different stakeholders, and people organised their own collection centres. A lot of people set up fridges and that kind of thing to collect and keep the badgers fresh in. We logged where the badgers were found, and usually that was done by people dropping a pin on a mobile phone and texting it or emailing it to us. There were a few times where I had to ring up and have long conversations with people while I was on Google Maps, and they'd say, oh, can you see if you just go along the road from the phone box and look to the left opposite the Feather and whatever it is pub, then that's where we picked up the badger. So, but most of them were by GPS, which I think logs them to the nearest 5 meters, something like that, which of course tells you nothing about where their sets would have been and the rest of the badgers in their colony would have been. I think we picked up pretty much all those badgers within 48 hours. And if it did take us 48 hours, we process the same time. If we can get them quicker than that, then sometimes we store them for a while before we, but no more than 48 hours again before we did the post-mortems on them. And we triaged those carcasses. So first of all, none of, well, I think 2 of them are frozen, but that's all. We wanted them as fresh as possible and as little damage as possible. So I did ask collectors to try and collect badgers like this rather than like this, and we did get a few like this, but on the whole, everybody was very, very helpful. It's no good having badgers which are damaged, because the bacteria that you find normally in the badger's gut and on their skin grows much faster than the Mycobacterium bovis. So it overgrows our cultures, and that gives you false negatives. So we wanted to make sure that everything was as sensitive and as clean as possible. We didn't do full post-mortems on these badgers. We looked for anything obvious. We checked that the main lesions were associated with road traffic accidents. The necropsies were all done in a special cabinet in the post-mortem room, mainly here at Nottingham, a few of them done in Liverpool. We had a set protocol, a set of tissues which were based on the ones that APHA, VLA have used in the past for their surveys of TB in badgers. They were stored overnight, and then the next day they were ground up in a category, or in a CL3, containment level 3 lab, which is the high containment. It's not the highest you can get, but it's the same containment you need for things like bubonic plague, HIV, and COVID now, of course. And then we tried to grow the Mycobacterium bovis that causes bovine TB from those cultures, and we did that on 2 different types of media, and we left them growing for 12 weeks. We checked them regularly to see if there was anything growing. So a lot of recommendations and protocols say 8 weeks or 10 weeks, but we thought we'd give them as long as possible. And one of the reasons for that is that we've done some work in the past on Vol TB, and we know that takes a lot longer to grow. So we just didn't want to miss anything out. And what we were looking for were colonies like this. And then when we got a batch of those, so it was worth doing, we extracted the DNA from them and did a PCR on those. So that is a PCR, so it detects DNA of a particular sequence, which is called insertion sequence 6110, which is associated with Mycobacterium tuberculosis complex. So it's not just Mycobacterium bovis, it's Mycobacterium tuberculosis complex. And I'll come back in a little bit to why that might be important. And then we batched up all of those positives. We spolytyped them to find out what strains they were, so that could be compared with the strains that were found in local cattle. And we also extracted DNA for sequencing. That hasn't worked quite so well, but we do now have 30 whole genome sequences from the badgers, and I'll share some of that with you later on when we get to it. So why do we use culture? We used culture because a culture positive means positive, because you can absolutely pin this down. Oh, you can pin it down as well as anybody can pin it down to being Mycobacterium bovis. Whenever you have a really specific test, and all these debates have been going on about TB and are now going about COVID as well, the more specific your test, often the less sensitive it is, and the more sensitive it is, the less specific it is. So we went for specificity. We didn't want anybody to be saying, oh no, it wasn't really TB, it was something else. But what that means is that while a positive means a positive, a negative doesn't necessarily mean a negative. So we might be estimating how much TB there is out there. On the other hand, that's probably true of all the surveys, whatever test you use in cattle. So using the jargon, high specificity but low sensitivity. I'm not sure it's really low sensitivity, it's just slightly lower sensitivity than if you did some other things. And I'll come back and discuss that in the context of results later. Right, results. We got 658 badgers, which we thought were worth sampling further, and 611 of them were not only suitable for post-mortem and sampling, but they weren't contaminated and they didn't have overgrowth of other bacteria on the cultures and so on. And we had good location data on them. We also had a few carcasses from Lincolnshire, South Yorks, and a few from Gloucestershire. I think this next bit's quite important. So about 40% of those badgers were collected by what I'm calling farmers, which is actually a range of people, but farmers, I think they're probably happy with that title. 36% from what I'm calling badgerers, and I don't know if you like the term badgerers or not. I don't mean it in an insulting way. It just brings together badger groups, wildlife trust groups, all those kind of groups. And I should say that badgerers and farmers are not mutually exclusive groups. And 24% from others. So over half of those others were us driving around to pick up the carcasses from farmers or badgerers. And if you look at things like age and sex of those badger carcasses, it was a roughly 50-50 male, female, and roughly 3/4 adult to 1/4 juvenile. And we're defining juvenile based on dentition and their coat color and that kind of thing. So it's an inexact science, but I'm emphasizing all of that to say that I don't think there are any biases in terms of who collected these badgers. And I think those kinds of figures for sex and age kind of reflect what people find when they look at live badger colonies and do what you might call proper ecological studies. So we haven't detected any obvious biases in the badgers that we collected. Nearly all of them came in the spring. We had a bit of an autumn peak as well. This is kind of expected because if you look at papers that people have published on, and your own experience, I'm sure as well, is that that's when badgers get run over. So it's kind of a combination of when badgers are active and when badgers are crossing roads and we're driving around. So it's those times of year when badgers are driving around when people are commuting to and from work. And in case I forget to say it, we're also working with some of the people in the ecology units at APHA on looking at the types of roads and the conditions of roads and bends and all that kind of thing to see if we can see if there are particular types of roads that lead to badgers being run over or whatever, and also comparing that with other surveys of badger densities and so on. I put a little chart down here to show that while overall who collected the badgers was pretty evenly spread, some counties it was more farmers, some counties it was more badger groups, and some counties it was even. Right, here's what you really wanted to know. So overall, 8% of those, now I'm trying to remember the number, I think it was 51, were confirmed positive for Mycobacterium tuberculosis complex. So I'll come back again to what that means in a second. Plus further badgers, and I forgot to calculate percentage beforehand, but it's between 3 and 4% on top of that were positive for other environmental mycobacteria. So as well as the Mycobacterium tuberculosis complex, which were responsible for causing TB in lots of different animals, including humans, there's a huge range of environmental mycobacteria. Some of them cause disease, some of them don't. And I'll come back at the end to why I think that's actually quite important, both in terms of diagnostics and possibly in terms of vaccination as well. The prevalence varied between counties. So on this graph here, I should say this, the tan coloured dots are badgers that were collected and negative, and the red coloured dots are the ones which were positive for TB complex. So just by eyeballing it, you can see that we had higher prevalence in Cheshire, Leicestershire and Warwickshire than we did in Derbyshire, Nottinghamshire and Northamptonshire. And I've got, so I forgot to put the figures in, but if you want to know the figures, it was 15% in Cheshire, but that gives you 95% confidence intervals of between 14 and 22%, I think. No, 9, yeah, it's 14% overall and 9 to 22%. You can be 95% sure it was in that interval. Leicestershire was 12.4%, which gives you a range of 7 to 20%. And Warwickshire was 9.8% of them were positive, which gives you a 5.4 to 17%. So big confidence intervals around that. And one of the reasons is because there wasn't much TB found in the badgers. You could reduce those confidence intervals as well by sampling 500 badgers per county, but we only had money to do 100. Nottinghamshire, Derbyshire and Northants were all between 4 and 5% of the badgers. That's all were positive. So if I now move away from anything statistical and start really annoying any statisticians who are listening in, then just looking at this map, I'd make a couple of observations. So in Cheshire, nearly all the positives are over here on the eastern side of Cheshire, which is where we found most of the TB before in 2014. And if you look at the rest of the map, it's kind of interesting that the badgers are all over the place. They're not right on the border with the high risk area in cattle. The other thing is you have to, and I guess I'm used to it, but the first time you look at these maps, it can be a bit confusing. The fact that it's all empty here doesn't mean none of the badgers or none of the cattle had TB. It's just that that's the high risk area and we weren't sampling badgers here. And the same goes for the low risk area over here. Right, let's go back to TB complex. So Mycobacterium tuberculosis complex is made up of 3 main types, subspecies, lots of different words you can use here. In the UK, there are even more globally. So there's Mycobacterium tuberculosis, which is the cause of most human TB. We weren't expecting to find that in badgers because it's difficult to imagine how badgers get infected with human TB, and we didn't find any. Mycobacterium bovis, or tuberculosis bovis, is the one that causes most bovine TB. But we also have microti, which is found in voles, and there's a picture of a field vole here with microti infection, vole TB, and you can see these lesions all over it. They tend to get quite big lesions, but they seem to be quite happily running around and breeding and having babies and all that kind of thing, despite being infected. So microti can infect badgers and cattle and people and cause TB in all of them. Right. So I put down here what's going on in a high risk area. Don't know, because apart from a few areas, nobody's really looked at badgers in the high risk area. What's going on in low risk area? Don't know. Very few people have looked in the low risk area. Remember that this is both an underestimate because we're doing cultures, but it's also a bit of a overestimate because that includes some of the microti, the vole TBs. Just a few more things on the on this sort of raw data, if you like. So I thought it was useful to look at, does the prevalence vary amongst different groups of badgers. So the only thing that came out of this which is statistically significant was that we had twice as much prevalence in males as in females. We didn't find that in the Cheshire study, but other people have found that in other studies of TB in badgers, and there's all kinds of reasons given for it. Everything from males are more likely to be fighting and transmit it that way, to maybe the males travel further distances, are more likely to come in contact with another infected animal of whatever species. So there's lots of reasons for that, but it was interesting that we saw that. But I think some of the non-significant non-differences, if you like, are also interesting. So I said before, roughly equal proportions of badgers overall were collected by farmers, badger groups and others, and the prevalence of TB amongst those was roughly the same as well. And I guess I tend to make a big thing about that, because I have been told in the past by some groups, oh, well, the people who are badger lovers will go and get you carcasses from areas where everybody knows there isn't any TB to make sure they're negative. And I've also been told by other people, oh, well, the farmers will go down to the southwest and bring you in lots of badgers that they've imported in there to make sure that as many of them as positive as possible. But we saw no evidence of that kind of thing. When we get to the spoligotyping, that's quite interesting as well. And I have got collections of DNA from all these badgers ready to go off, and we'll look at the population dynamics of those badgers. So we should be able to spot anything suggestive of somebody bringing in badgers from outside their area. The other thing here, and this is affected by the fact that nearly all the badgers were collected in the spring, but for all that, there was no obvious difference in the prevalence of TB at different times of year. And what that suggests to me is that if you wanted to save money and do this again, you would just do it in the spring months. And the thing down the bottom, and I'm not making a big deal out of the differences or the similarities here between the prevalence in juveniles and adults, except to point out that actually we did find quite a few juveniles infected with TB. So this isn't, that might suggest that there's insect transmission. I don't know. It kind of fits though, doesn't it, with some of the vaccine trials, which suggest that if you vaccinate adults, the biggest effect is on the following year's cubs. Don't know. I just thought that was interesting. Right. Here's the map with negative badgers and positive badgers on it. Here's the map with the positive badgers on it and cattle breakdowns or cattle outbreaks in exactly the same time period as we collected the badgers. You could argue, well, what you really need to look at is the cattle before to see if they were the source of infection, or the cattle afterwards. But actually, you don't answer those kinds of questions by doing that. And I just think it's unfair. So for all its faults, we're just going to compare what we found in the study time in cattle and badgers. Right, so I'm now going to be very naughty and unstatistical again and just make some observations on this map. So the first thing is up here in East Cheshire, we found badgers and cattle with TB. So that's the same as we found in 2014. These areas outlined in red are areas that I've picked out because we got lots of badgers from those areas, but none of them was positive, but there's quite a bit of cattle TB in those areas. The area in green I'm highlighting over here, we had a little cluster of TB in badgers, but not in cattle. That's partly, of course, because there aren't very many cattle in that area. And before everybody gets really excited, there's an area down here in orange where there's lots of TB in the cattle, none in the badgers. But one of the reasons, at least, why there's none in the badgers is we didn't actually get any badgers from there. So neither farmers nor badger groups managed to get any badgers from there. And I'm afraid I didn't notice that till we started putting the grants together. Otherwise, I might have taken a drive around there and seen what I could get. So I guess when we started this out, we thought there were three scenarios. One of them is you'd find lots of TB in the badgers, none in the cattle, and that would mean that badgers were driving the epidemic. Another one was lots of TB in the cattle, but not in the badgers, and that would mean that cattle are driving the epidemic. And the third one was we'd find a complete mixture. And actually what we've got is all three of those outcomes in different areas. So it's not a simple single story. And I guess in retrospect, that's kind of what we should have expected. Right, so I put in here a but. Remember, the badger results are MTC, Mycobacterium tuberculosis complex, not necessary, M. bovis. And one way of doing that is to spoligotype and try and identify, characterize the positive isolates that we got out of badgers in more detail. So all of our isolates have been spoligotyped, and like I said, about 30 of them we have whole genome sequences on. So I'm just going to talk about the spoligotyping to start with, and then I'll just mention something about the sequencing. So the spoligotypes, the strains on here are color coded. And that's the same map for the same period for spoligotypes in the cattle. Right, let's go back, because otherwise you're going to go boss-eyed trying to look at two maps at once. So I suppose what I wanted to point out from this map to start with is over here in East Cheshire, the most common spoligotype in the badgers was spoligotype 25, which is the same one we found in the past, and as we'll see, is the same one that you find in cattle. But we have a few where we have positive badgers in Derbyshire and a bit in Nottinghamshire. They're often spoligotype 25 as well. We also picked up some spoligotype 17s in Cheshire badgers, which we didn't find in 2014, and a few spoligotype 17s down here. And all of these yellow ones are just a bit weird. So we either couldn't work out what the spoligotype was, or it's a spoligotype which hasn't been found very commonly in cattle. The purpley ones, or lilac ones, are probably Mycobacteria microti. So these are the Vol TB samples that we can probably should take out of the map. Okay, so let's go back, look at the cattle, and what I'm going to do is click that, stop me looking at the badgers for a second. So the tan on this map means cattle outbreaks where there was no spoligotype available at the time. And in Leicestershire, because I'm talking largely to Leicestershire at the moment, you had a fairly new epidemic in cattle going off, and very few of the isolates that came out of cattle, if indeed they were culture positive, were spoligotyped. So actually most of those were probably skin test positive and were not confirmed by isolation. Doesn't mean the cows didn't have TB, it just means that, well, there are all kinds of reasons why you might not get it out of those cows. So if you look at the colours on here, there's the 25, lots of grey ones up there. More red ones than we would have seen in the past. This is the spoligotype 17. And down here in the kind of Warwickshire area, then we have the green ones, which are spoligotype 10, with a smattering of blue ones, spoligotype 9. So I thought I'd just talk about that a bit more, and the regionalisation. If you go back to that early map I showed you about where you get different spoligotypes. So spoligotype 25 is traditionally found in this kind of area, particularly in Staffordshire and so on, and that seems to be the spoligotype which has been undergoing epidemic spread into Cheshire, or at least East Cheshire, and into Derbyshire, and I'm not quite sure where we have a hole here, but into Nottinghamshire as well in cattle. Spoligotype 17 started off down here and has spread gradually in that direction. So that's why there's a few of them being picked up, and up the Welsh borders and into West Cheshire. So more and more cattle which test positive in West Cheshire are spoligotype 17. And I'm not quite sure what else to say really. So basically, if the badgers fitted the same pattern, we would expect lots of spoligotype 25 up here, lots of spoligotype 10, the green ones down here, and maybe a smattering of other colours, because who knows what's going on in terms of long distance. And I'll talk about other reasons for possible long distance transmission other than just cattle movements in a bit. So now I'll try and, oh, that's my little arrows. It's a bit like the old Dad's Army thing, isn't it? Right, so let's put the cattle back up, the badgers up again. So what I just said really, East Cheshire, spoligotype 25 in both cattle and badgers, with for the first time a few spoligotype 17s turning up in badgers, and both of these I think mapped onto farms where there are spoligotype 17 outbreaks in the cattle. You can argue whatever you like about directions of travel and transmission and that kind of thing. Over here in Derbyshire, as the Derbyshire listeners will know, very few badgers with TB, but those that do tend to be Spolyga type 25. And we've got a few Spolyga type 25s over here on the other side of Nottinghamshire, which kind of associated with, it's the A1, isn't it, going up there. And then the other obvious thing is that down here we have a whole smorgasbord, Jackson Pollock, whatever you want to call it, of different coloured dots all over the place. But what we don't see, which we see in the cattle map, is lots and lots of these green Spolyga type 10 infections. So there are problems with Spolyga typing. But if we believe what we've got so far, then we've got different stories again in different edge counties. So I'm probably over-interpreting the data now, and it won't be worded quite like this in the paper. But we have a relatively high prevalence of TB in both cattle and badgers in East Cheshire, and it's nearly all Spolyga type 25, and a lot of them are co-located. So it's probably the same epidemic, same as we found in 2014. We've also found increasing numbers of Spolyga type 17, or rather APHA found increasing numbers of Spolyga type 17 in cattle, and we've seen a small number of those Spolyga type isolations from badgers, and where we've seen that, it co-locates to Spolyga type 17 in the cattle. So I think we have the same epidemic going on, but you can't still say which direction it's going on in, because in some ways that area of Cheshire is a high risk area and it's all out of control. Derbyshire and Nottinghamshire, increasing epidemic of Spolyga type 25 in cattle, especially in Derbyshire, but a low prevalence of sporadic Spolyga type 25 in badgers, often co-locating with cattle. But I would say, and I think our paper will say, there is carefully worded, no evidence support badgers driving the cattle epidemic in Derbyshire or Nottinghamshire in the period where we were doing the testing. Leicestershire and Warwickshire, I'm afraid you're a bit of a mix and a bit more complicated. So we've got TB in the cattle and the badgers, but if we believe the Spolyga typing, it's kind of difficult in Leicestershire because many of the cattle isolates at this time were not Spolyga typed, and the badger isolates are a bit weird and all over the place. I've got to think of a nice way of saying a bit weird when we write the very final version of the paper. And in Northamptonshire, at the time of the study, there was very little TB in either the cattle or the badgers, with little evidence of similar Spolyga types or co-localization. So my conclusion is it's a mess in Cheshire. In Derbyshire and Nottinghamshire, we need to protect the badgers from getting infected from the cattle epidemic. And in Northamptonshire, we need to make sure that we keep TB out of both species. And in Leicestershire, not quite sure what's going on. But again, we don't really have, lack of evidence is not the same as evidence of not, but we don't have any evidence of co-location of infection in badgers and cattle with the same Spolyga types. So we don't have any evidence that either one is driving whatever's going on in the other, in those counties. David asked me if I could do some bigger maps for Leicestershire. So that's the overall map with the Spolyga types in badgers in. This is blowing it up to Leicestershire. And frankly, it tells you very little, because as soon as you start blowing these maps up, your eye is drawn to the whole areas where we didn't get any badgers at all. And I've already said, so this is a kind of an interesting area over here where we had a cluster of badgers in an area where there are very few cattle TB outbreaks. So 2 of them at least are microti, and most of the others we don't know what Spolyga type they are. So some of them could be microti, they could be something weird, I don't know. When I've talked to both badger and farming groups for the Vale of Beaver, then and I'm trying to work out exactly where it is, but there are a couple of Spolyga type 25 ones, and I think that might be one of them, where it where there's a co-location of Spaliga type 25 in cattle and in badgers, and there have been consistent outbreaks in cattle in those areas. So it may well be that we're seeing the start of something like a mini Cheshire going on in some of those areas. So we have a hotspot in both cattle and badgers. But what there is not any evidence of is an epidemic of Spaliga type 25 going, let me go back again. There's no evidence of an epidemic of Spaliga type 25 going whooshing through badgers all the way over to here. These are sporadic things, and quite a long way from the main epidemic in badgers, although not necessarily a long way from the main epidemic in cattle. Right. See if I can get through this again. Keep an eye on the box. Right. I want to distract you now with some other, what I think are really interesting things. So I said I would come back to these other non-tuberculous mycobacteria. So up here we have a little family tree. Let's see if I click on it. Oh yeah, you can just see. This is all the different mycobacteria which have been sequenced and characterized that you find in the world. These little red ones here are the Mycobacterium tuberculosis complex. So you can see that we're dealing with a very small minority of mycobacteria. In as far as we have characterized the non-tuberculous mycobacteria that we've isolated from about 3 or 4% of the badgers, well, they've been quite interesting. So I've highlighted some of here. There's Mycobacterium non-chromogenicum. I hope you're taking notes on this, Ian, because we discussed this briefly yesterday, and you said you hadn't heard of most of these. So take notes. Mycobacterium vaccae. I've just put tuberculosis in there so you can see where it was on this kind of tree. Avium and paratuberculosis, Johne's disease in cattle. Malmoense, and Hasearchum, both of which are human pathogens and can be quite nasty and cause quite nasty disease. One of our pathology residents has just finished his MRes, his Master's in Research, and he looked at the histopathology of a whole load of these badgers. And he found that there was Ziehl-Neelsen's acid fast, that's to say mycobacterium-like bacteria in microscopic lesions in both the badgers that we confirmed as having TB and in these other badgers which don't have TB, which have mycobacterial infections. And there was no real difference in these microscopic lesions. So what we're going to try and do is characterize these further and characterize a lot more of them. And the labs were all closed down because of COVID before another MRes student could finish that up. She prepared all the samples and then it all went wrong for her. But the reason I'm making a bit of a fuss about this is if you're in an area where they were, say, 50% of the badgers had TB, then an extra 3 or 4% of them that have this kind of mycobacterial or these kind of mycobacterial infections makes no difference. If, however, if you're in an area where there were 3 or 4% of the badgers infected with TB, and you used a diagnostic test which just said whether or not they had a mycobacterial infection, you would end up thinking you had a prevalence of 6%, but half of them wouldn't actually have TB. So there is a diagnostic possible problem here. And of course, I don't know what rate of infection there is with other enteric, with other non-tuberculous mycobacteria in other areas of the country as well, or in other countries. So that's one possible problem is with diagnostics. The other one is that in humans, certainly if you're as old as me, but actually once you get to the age of 30, it's generally reckoned it's not worth immunizing you against TB. There's no point giving you the TB vaccine, BCG. And the reason for that is it doesn't work very well. And it's thought that the reason that it doesn't work very well is that by that age, you have been infected with a whole load of these non-tuberculous mycobacteria. And while they're different enough that they don't protect you from TB, they're similar enough that they protect you from the vaccine. So you would expect badgers to have quite a lot of these kinds of infections. And I'm just slightly worried as to whether or not that might affect vaccine efficacy in badgers. So I don't know whether any of that is good news or bad news. I'm not going to say too much about whole genome sequencing, except to say we've got full whole genome sequences from 30 of the isolates. On the whole, the spot oligotypes you infer from those sequences are the same as spot oligotypes we found by spot oligotyping. There's a small bunch of them where the spot oligotype is different, but it's still a nonsense weird spot oligotype. And there's a few of them where the predicted spot oligotype from whole genome sequencing says it's one of the more common strains, so a 17 or a 25 or something like that. There are lots of reasons why these two approaches might not join up, but we haven't got to the bottom of what all of those reasons were, or which ones which approach you believe. So the PhD student's still working on that. If you draw a map and compare that with a phylogenetic tree of these sequences, then it looks as though there's quite a big difference between, there's a splurge of sequences which are closely related, which are in the northern half of our study area, and a splurge of sequences not quite so closely related in the southern half of our study area. So that kind of fits with a spot oligotype type 25 across the north and something else along the south. So it's not one infection in badgers, all the ones not one strain of infection, all the badgers. And I guess the other thing is that all the ones we've looked at so far are indeed Mycobacterium bovis, although there is some difference between published sequences of Mycobacterium bovis and the Mycobacterium bovis whole genome sequences we've got, which are still to be worked on. Right. In all of this study, we only had 2 badgers with widespread lesions and 2 others with obvious lesions. That's 2 TB positive badgers with widespread lesions and 2 further with obvious lesions. What that means is that something like 90% of the badgers, and we saw this exactly the same picture with the Cheshire badgers, don't show any obvious clinical signs or obvious lesions from the outside. Interestingly, one of the badgers that had an environmental non-tuberculous Mycobacterium infection had very similar gross systemic lesions to the badgers that had proper TB, and another one of the environmental Mycobacteria had systemic lesions, but they didn't quite look like proper TB. So you kind of thought, oh, I wonder if that one's TB. Oh no, I'm not surprised it's not really. But my bottom line on this is anybody who tells you that you can spot a tuberculous badger at a distance is talking out of their nether regions. We've got some stuff I'm not going to go into about the distribution of which tissues were positive and which were not so often positive in the badgers, except to say we'll put all of that together, and I think it could be quite useful in terms of doing surveys in the future, because you could cut down the number of samples that you need and still get the same kind of results, which would save everybody a lot of money. This is an early, quick and dirty analysis of, I think, 30 TB positive and 30 TB negative badgers, and looking at all lesions, not just ones which look a bit like TB. And the thing I just want to concentrate on here is the grey bit, the no lesions. So basically, TB negative badgers very often had no lesions of any sort, apart from those associated with being hit by a car or truck. But the ones with TB, far few of them had no lesions. Now, whether this is just chance, or whether this suggests that badgers that are infected or have some other disease are more prone to get TB, or whether it suggests that ones that have TB, even though they don't have obvious signs of TB, are more prone to getting other infectious diseases, I don't know. But we think it's something that's worth looking into in more detail. And the pathology student who is doing this is doing exactly that at the moment. Because of that, we're also looking for other infections. We found that 20% of the badgers are excreting salmonella in their poo. And just to show that we're not totally incompetent when it comes to sequences, we have whole genome sequences from all of those, and we know their antibiotic resistance profiles. So just as a little taster for all of that, because we're still doing a lot of the analysis on that, the clever phylogenetic stuff, the badgers from Cheshire had very little antibiotic resistance, but those from the East Midlands were quite dirty and had a lot of multi-drug resistance. Don't know what's going on there really. I've had a couple of veterinary students do their projects looking for gamma herpes virus and retroviruses because some people suggested they might be associated with a higher risk of infection with TB. But basically nearly every badger is infected with gamma herpes virus. And we didn't find any evidence of retroviruses in badgers at all, which is quite interesting, except maybe the briefest hint that in their evolutionary past, they might have had a retrovirus, but it was kind of a similar sequence to that found in a whole load of mammals. So it goes a long, long way back. We've had other students looking at E. coli and antimicrobial resistance, and we need to kind of finish that off and get on with it and publish it. And we found all kinds of other infections in these badgers, leptospirosis, emontia, whole load of other things. Quite a lot of the badgers had nephritis, quite a few of them had hepatitis, and some of these things seem to be associated more frequently with TB. Most of them have no relationship with TB, and there's a few of them, very few of them, which you never find in the animals which are TB positive. And again, whether that's chance or not is, that's up to the statistics and longer term studies to find out. And then I put this one in case Debbie's watching, because we also decided to look at the livers of all these badgers. I don't know what made me do it, but I said, oh, let's freeze down all their livers. And then there's somebody here who's really good at going looking for heavy metals, as in metals with rock bands and that kind of thing, in all the livers. And we looked for about 50 different, we could call them pollutants, I suppose. So I've just put one example up here. These are the British Geological Survey maps of where you find cadmium and where you find lead in England. And the orange bars here are the average cadmium in that case and lead levels in the badger livers. These averages for the counties cover up a huge wide range, not in the other counties, but in Cheshire, in Derbyshire rather. The orange lines are Derbyshire. Huge wide range where some of these badgers had between 50 and 500 times as much cadmium and lead in their livers as an ordinary badger would. We found a few badgers elsewhere in the country, I should have put a map in really, which had quite high lead levels as well. So we're going to look at them in more detail, because apparently the lead that used to go into petrol and paint was all imported from, I think, Australia. And that has a slightly different mix of isotopes and therefore different molecular weight from the lead that you find naturally in Britain. So we should be able to distinguish badgers which have got high lead levels because they've been living in areas where there's lots of lead mines from those that have got high lead levels because they live near big road interchanges or at an old garage site or where somebody used to have a paint store or something. And finally, I think I've been wittering for far too long. I just want to come back to this, and I've talked about this at some other meetings as well before, but it's changed a little bit since the last time I mentioned this. How well does Mycobacterium bovis survive in the soil and other stuff like manure and so on? And the answer to that is it's known it can survive in the right conditions, weeks to months. It doesn't survive very long if it's exposed to UV, if it gets too dry and things like that. But if it's the right kind of conditions, it can survive for quite a long time. And the question I was asking about this is, can this be helped by protozoa that live in the soil? And the reason I was interested in that was I had a PhD student who was working on acanthamoeba in water. And the reason that's interesting is it can cause eye lesions in people who wear contact lenses. And at the time I was asking this question, there were, I think, 2 papers out there that looked at acanthamoeba and bovine TB. So acanthamoeba in the wild can be full of all kinds of different mycobacteria, many of which actually grow in the acanthamoeba and are protected by the acanthamoeba. So one of these papers on Mycobacterium bovis said, no, it can't grow in acanthamoeba. Acanthamoeba protozoa just eat Mycobacterium bovis and digest it. And another one said, well, if you feed them with BCG, the vaccine strain, they just eat it. But if you give them a wild type bovine TB strain, that can grow inside the acanthamoeba. So this is 2017, but I only recently came across this paper, and these people looked at a whole load of acanthamoebas and other protozoa from the wild and looked at their ability to host Mycobacterium bovis. And basically Mycobacterium bovis was able to grow in at least 5 different species of protozoa. And what's more, these protozoa can form cysts which can survive for years in the environment. They didn't let it go for that long, but they found they could survive. Mycobacterium bovis bacilli could survive in a cyst for a couple of months. So they said over 60 days, that's when they stopped their experiment. And furthermore, and I've chopped it off here, they were able to then take the Mycobacterium bovis out of them, infect mice and cause TB in them. So my reason for thinking all of that is quite interesting is if Mycobacterium bovis survives really well in the soil and even better in Acanthamoeba, that could explain some of the long distance transport. It could also, because on mud, on trucks, on the fur of animals, on the feather of birds, on keen people going to watch badger sets, farmers going from one farm to another. There's no blame associated with this. It's just that it could be moved around short distances and long distances really easily, and it could survive in the environment for a really long time. And I'm not sure that anybody's really thought about that in terms of the epidemiology, except I go back to what I said in the very first slide, that a lot of the models of the epidemiology of Mycobacterium bovis use the term environment. And if you look at those models, it's difficult to know whether you would get exactly the results if you really meant environment as opposed to whether you really meant badgers. I'll shut up now, except to say thank you very much and just re-emphasize there were loads and loads of people who are involved in all of this. I'll stop sharing. Have I stopped sharing? I don't think I have.
Yes, you have, yeah.
Excellent.
Thank you for that. Okay, Ian.
Who's awake?
Lost Ian.
I'm back.
Here he is. Okay, we'll pass over to you now then.
Hello. Well, first of all, I'd like to say thank you very much to Malcolm. It's very important that we have independent scientists in universities that are prepared to speak the truth, because there's a lot of government scientists don't have that option, and it's very important to remember that. So the first things I'm going to talk about are, I'm not going to I'm sure your brains are pretty frazzled after all the amazing stuff that that Malcolm has shown you, but there's a couple of things I would like to show you. So I'm just going to share my screen, and I'll just talk around this. Okay, can you see that?
Yes.
Okay. So there's lots of badger killers out there who are now, their latest strategy is to say, oh, badger killing's working. Badger killing's really good, or we're getting really improved results in cattle because we're killing badgers. So just to show what a lie that is. So this graph is the incidence rate of cattle tuberculosis in the areas of Great Britain, England, Wales, and Scotland. Scotland doesn't really have a problem. So here we are. We're in March 1986, 1996, and we're going along. This is, the brilliant idea of killing millions of cattle and sheep because of foot and mouth disease. After that, they suspended all the controls for moving cattle around Britain. So you could move cattle from the low risk area, from the high risk area in the West Country, all the way up to Cumbria, all the way up to Scotland, anywhere you like, without testing them, because there's so many killed that they wanted to suspend all the controls so that people could restock. So this caused this massive increase in around 2002. So that's carried on going. So in 2013. You get badger killing starts. So here it is, badger killing starts. And you can see in England, it hasn't really had a lot of impact. The disease has carried on, this green line carries on. In Wales, where they've not killed badgers, they've had no systemic killing of badgers. They have killed the odd one or two, but they haven't had mass badger culling like we've had here. You can see that Wales drops off. You know, they're a bit more progressive than England, and they're working. So that's not really true that the badger killing is working. I mean, not only that, even if this were to be coming down, then they've done so many other things. They've changed the testing that they do for cattle, and we'll come back to testing in a little bit. They've used severe interpretation, which means with the skin test, you pick up more of the cattle that the normal interpretation was missing. They've started using gamma testing, which is picking up a lot more infected cattle that, again, the skin test is missing. They've done a lot of things that are helping to bring down TB. I mean, TB, as you can see from this graph, has been spinning out of control for the last 20 years. But any improvements you might get, and let's face it, from this graph, there's precious little in England. There are many, many factors that are being used there. It's not just killing badgers. So that's the first graph I want to show you. That's what's going on in the cattle disease. So the next thing I want to do is to get you the next thing up. Can everyone still see that?
Can't see anything at the moment.
Okay, hang on.
Yes, we can see that now.
Okay, so that was the cattle disease, and now we're moving on to the complications of badgers. So what we're looking at here is, it's still the cattle disease, but we're looking at the cattle disease in the 3 longest running areas where they've been killing badgers. So let's look at Gloucestershire for starters. Okay, so Gloucestershire, they've been culling badgers since 2013. So you can see it was quite high. This is the incidence rate of new breakdowns with TB in cattle herds. You can see it's quite high in 2011, and it's coming down. So then they start culling badgers, and just look at the solid line for the moment. So they start culling badgers, and it goes up a bit, and then it starts going. It plateaus, and then it goes up again. There's a government paper which looks at all these areas, and it takes the data up until 2017. You can see that after 2017 in Gloucestershire, there's a big hike going on here, and that's not included in this data. So this is the incidence rate. There's also prevalence data, which looks even worse for this. So it really goes up in Gloucestershire and also in Dorset. Somerset's a bit more complicated. But the reason I want to show you this is for 2 reasons. So firstly, there's the fact that it's not really working. It's going up. Secondly, the dashed lines here, this is called herds in existence. So these are herds that are in the area of badger culling, but they're not fully exposed to the full effects of badger culling. They might have moved in later. They might not have been in the area for the whole time, but these are called herds in existence. So what you'll notice is that, to start with, the herds in existence were doing a bit better than the cohorts, and the cohorts are the ones that have been exposed to badger culling for the full period. You'll see that they're doing slightly worse. As time goes on, that reverses. So that the cohorts at the end of the study are doing worse than the herds in existence, which haven't had the full impacts of badger culling, which is a reverse from what they started. So in Somerset, it's the same. If we look at Dorset, Dorset didn't start culling till 2015. But again, there's a similar pattern going on. I haven't actually done the graph for prevalence, which is the percentage of herds. I'll maybe try and do that as we go on. I should have done that before. But what they're showing is, one, it's not really working. The government has published a paper up until here saying, oh, it's all fine and dandy, everything's working. But we've got the data up until 2018, but the government don't want to look at that. They want to concentrate on this Downs paper. They don't want to look at the fact there's been a really big 130% increase in Gloucestershire. They really don't want to talk about it. So these farmers in Gloucestershire, they've been culling now for 7 years, and they haven't received, there's no benefit at all for culling badgers. So I'll just see if I can stop sharing this. Has that worked? Am I back to not sharing?
Yes, you are. Thank you for that.
So what's the reason for this failure of the bTB policy? Why is it not working? So firstly, the government, they want you to believe that it's all about badgers, that badgers are the cause of it. We're killing badgers and it's getting better. As Malcolm showed, there's a lot of that's very equivocal, and particularly in some areas, there's no evidence for the badgers driving the area at all. Everyone knows that cattle will give TB to badgers. That doesn't mean that badgers are causing the disease in cattle. So what's the main reason for the failure of this policy? You saw it was going up and up and up. In Wales, it started coming down, even though they've not killed any badgers systemically like they're doing in Britain. And the main reason for that is because the skin test they're using, it's all about testing. The skin test that they're using in England just isn't working. So what they call that is the single intradermal comparative cervical tuberculin test, or SICT. And that's where they inject some tuberculin. One of them is tuberculosis, and one of them is the avian form. And then they compare the two to try and work out, was this animal exposed to TB? So it's an immune reaction. It's not actually looking, as Malcolm was doing, to see if there's actually tuberculosis, M. bovis is there. So what I call that test is the silly Edwardian skin lump test, because this was invented, well over 100 years ago, and it's what this entire policy is based on. What the government don't want you to know is that this test misses half of all infections. So that's a sensitivity of around 50%. Two big government studies published in 2018 show that this test that we're basing the whole policy on is missing 50% of infections. Now, no, that's incredible. So that means if you're testing a herd and there's 10 cattle that have actually got TB, you're only going to pick up 5 of them. And then after a while, those cattle will test clear, they won't be showing up, and the farmer will be given a certificate saying, oh, you're officially TB free. And actually those 5 infected animals are still there. So 6 months later, a year later, the herd goes down again with bovine TB. And the farmer goes, well, it must be the badgers. The government says it's the badgers. The local vets told me it's the badgers. It must be the badgers. We have to kill badgers. And what the reason is this ridiculous Edwardian skin test. It just doesn't work. We've got a lot of molecular tests. We've got molecular tests that can tell whether the animal's infected or whether it's got TB. But the government doesn't want to use it. The government wants to carry on with the skin test. There's a whole system set up to use the skin test. And that's the main reason this policy has been a disaster. Even more worrying is they're using the same 50% skin test when they screen cattle for moving. So at the moment, you can take cows, one cow, a whole herd of cows, and move them from the high risk area to the low risk area, and you use the same skin test. So what that's doing is, if you want to move 30 cows, 2 of them have got TB. You use the skin test, they're only picking up 50% of the animals. It may well miss those 2 animals. And if there's only 1 infected, it's very likely to miss it. So then these animals travel up to the low risk area, say up to Cumbria, and they've not been screened properly because this test is only picking up. I mean, even the government admits this is a risk reduction measure. It's not an effective screen to stop infected cattle moving. So in Cumbria, for example, very similar testing protocol was used. Animals were brought over from Northern Ireland. They were carrying the 17Z Spoliger type of TB. And lo and behold, they were infected. They weren't screened properly. And then you've got an outbreak and a little epidemic in Cumbria. And the immediate response is, oh, it must be the badgers. We'll kill the badgers. Even though the government knows full well it's because the screening test didn't work. So what that's meaning is that this skin test is not only not picking up the disease in cattle, so it's living a huge undiagnosed reservoir, which is going to be probably around about 20 to 30,000 animals left in the herd, even in the high risk area, without looking at the whole country. And then they're using the same test, and those animals are then spreading around the country. So it's, you know, it's inevitable that the disease is going to spread everywhere in the UK if they carry on using this test. I mean, they are using other tests, they're using gamma. They're very, very resistant to using a test like actiphage, for example, which is very sensitive, very specific. It's using very similar tests to the ones Malcolm was using. It's a PCR test, the polymerase chain reaction test, with a phage virus that's going to puncture the actual tuberculous bacteria, and it's going to detect only the DNA that's from M. bovis. So it's very specific. And it should be being used, and the government's not using it. So, as you saw from Malcolm's work, there's the badger infection overlaps cattle infections. It doesn't mean that the badgers are driving the epidemic, and there's a lot of evidence suggesting it's not the badgers at all, that it's the cattle. If you've got a massive undiagnosed reservoir in cattle herds and an ineffective screening test, you're going to spread it everywhere. So the chief veterinary officer has said that Christine Middlemiss says she's got conclusive proof that it's the badgers causing problems in areas like Lincolnshire and Cumbria, which are the low risk area. Cattle moves in, which has got infection, it starts an epidemic, and then they kill the badgers. Well, we'd like to see that. We're not at all convinced that that's true, and we'll be taking that further. I mean, as Malcolm said, there's no grounds in many areas to support the fact that badgers are driving the epidemic. So I didn't find out I was doing this talk till yesterday. So I'm just going to give you a little snapshot of what we're doing. So some of you may have seen a big group of us, including very senior vets and Jane Goodall and Chris Packham, wrote to the Prime Minister saying he should intervene in Derbyshire because, for all the reasons I've outlined, there's no evidence that no hard evidence that badgers are the cause of this, and a lot of evidence showing that it's cattle. And the fact that last year, because Carrie Simmons, his partner, is a vegan activist and likes badgers, pressure was put through her to persuade Boris that he should pull the Derbyshire cull zone last year, which he did. It was a political decision, not a scientific one, and he did that. So we said to him, well, you've done it before, you can do it again. Why don't you just pull the cull? These are the reasons. And obviously we didn't really expect that he would, but we knew that it would embarrass him. He got on the front page of the Vet Times, main news article in the Vet Record, both of them published it. It was in the independent newspaper. It got far and wide, which obviously makes the badger killers very angry that we're getting all this press. So we wrote this letter to the Prime Minister. The British Cattle Veterinary Association got very, they clearly rushed off a letter, sent it to the Veterinary Record and the Veterinary Times, and made quite a lot of mistakes. So we've replied to that letter, and we're scathing about them in a way that vets usually aren't scathing, to other vets. We accuse them of trying to manufacture consent for the badger cull by presenting misleading information systematically, which is an unusual thing for vets to say. And I would go further than that. I think that what's going on with badgers, you've seen a little bit of the evidence from Malcolm. It's a campaign of systematic scientific fraud that's going on to manufacture consent for this policy when there's no evidence for it. What they're hoping is that the cattle measures that they put in place will bring tuberculosis levels in cattle down, and then they can claim it's all to do with badgers. But that's all happening a bit too slowly for them. And it's not quite working fast enough, and it's clear by comparison with Wales, we've got controls there, and you can see that it's working in Wales and it's not working here. So why would they want that to happen? That's a good question. But if you look at the budget for TB in the UK, bear in mind this is a disease that doesn't cause much. There's a handful of human cases a year since we pasteurized milk. So we've got this system set up. It's effectively a nationalized disease, and it's got a budget of 100 million pounds. So if you're getting a big slice of that pie, you might not be very happy about the solution being solved. You might be quite happy that the gravy train's running. So you might not really want it fixed. You might want a silly Edwardian skin lump test rather than a proper molecular test. so that the gravy train keeps going on. And I don't think that's really acceptable. I think we have to look at the science. So we need people like Malcolm who are going to explain what their findings are. And you've got to remember there's a lot of people in the State Veterinary Service, the APHA and DEFRA, who'd also like to tell the truth, but they're not. They're restrained from doing so. They're not allowed to talk to the media. And what's put out by APHA is very much censored. I used to work for MAF doing research on BSE. I was part of the sort of cutting edge team that was, you know, it was a new disease. I was working with the guy that discovered the disease, Gerald Wells. I was very young at the time and my work was censored, which means it came back from the senior vets, the deputy chief vet and the chief vet saying, we're not, you know, you can't publish this. You know, you either take this out. It had black lines through the paper. You either take this out or you're not allowed to publish it. And that was when the disease jumped from cattle into cats, which is very significant because it went from a herbivore into a carnivore. Lots of people were eating meat. And we wanted to say this increased the risk to people, and it came back with black lines through it. And that's going on a lot of the time. People are trying to say the right thing, but they're not allowed to. So, you know, we need to get on top of the disease. We can't let it carry on like this and, you know, blame it on badgers that aren't responsible for it. We need to start using proper testing. And the fact people are making a lot of money out of it is not a good enough reason to keep this system going on. So as I said, I think this is systemic, scientific and financial fraud, and we need to do something about it and not blame badger. Thank you very much.
Thanks, Ian. Okay. Right, we've got everybody back. And we'll start working our way through some questions. We've got quite a few questions coming into the Q&A box here. So we'll, as I said at the beginning, we'll work through as many as we can. I'll try and find people and allow you to turn your video on and your speaker on if you would like to. So our first question comes from Hannah Meadows and Catherine Cooksley. So you should be able to talk if you would like.
So if that's to me, then the answer is yes, but not in the study. So the actiphage test that Ian was just mentioning was developed here at Nottingham by some mates, colleagues, whatever you want to call them, in another building, but we share all kind of stuff. And I know they've been using their test in camelids because in camelids, the skin test doesn't work very well and it's really difficult to diagnose TB. And the other problem with camelids is they tend to get an enteric infection rather than a respiratory infection. And the reason that's a problem is twofold. First of all, it all comes out in their poo and contaminates the environment. And the other one is they spit it directly into your face. So if you get spat at by an alpaca, they're regurgitating stuff from their small intestine. So it's doubly bad because it smells horrible and it's could be full of TB. But we didn't specifically study that in this study because all we were looking at was, you know, the question was badgers in the edge area or the northern edge area. And I was going to say something else there, but my mind's completely gone. Can somebody say something intelligent? Bail me out, Ian. This is what cheers.
I agree with Malcolm. That's all I can say.
I try not to agree with him. He gets me into trouble.
Okay, so if you're unsure of the questions, if everybody watching, if you just open up the Q&A box at the bottom, and everyone should be able to see the questions that have been submitted.
Sorry, I should have summarised the question, shouldn't I, before I went off on a
just in case people are missing it. So the next one is from a lady called Anne. So Anne, I will also allow you to join us, and you can turn your video on and your speaker if you wish. So it's quite a long question. you-
Right, what it does is it switches you off and then switches you back on.
Okay.
Yeah, that's what happened. I raised a question about the TB vaccine for children. And a couple of months ago during COVID, I had occasion to check when we'd started vaccinating and what for. And we started in the 30s with diphtheria. And when I got down to BCG, I found something I found rather curious, because it explained to me why there was a higher prevalence of medics catching COVID who were from minority ethnic backgrounds. And what the BCG vaccination advice to medics is, if a child has a parent or a grandparent who was born in a country where there is TB prevalence, then they should be given the BCG vaccination. And that suggested to me that there must be some kind of inherited disposition. And I wondered if the origins of TB in cattle actually goes much further back.
So I might be able to answer a bit of that, because I've had to do some reading around BCG and who should be vaccinated and who shouldn't, because it's a long-standing issue in veterinary schools as to whether or not students should be immunized or not. And I think the answer is that it's nothing to do with genetics and inheritance or predispositions. It's simply that those are countries in which there are very high rates of TB in humans, and as in cattle and badgers, it tends to be a lifelong infection if you get infected with it. And so the idea was it was more likely to be passed on from one generation within that family. And there are probably all kinds of other things I'd rather not get into as to why that might particularly happen, but probably, you know, the same kind of reasons in terms of socioeconomics and where people live and large families and all that kind of thing that lead to higher rates of COVID in some parts of the country.
Yeah, but
so I think that's the answer. I think that's the simple answer to it. And on the whole, the medics don't like immunizing against BCG, because immunizing with BCG, because we don't have a lot of TB in the UK. So I'm trying to remember, I think it's only in London, or certain boroughs in London, where they routinely vaccinate nowadays. It's quite difficult to get a vaccination with BCG nowadays, unless you're in certain boroughs in London, or you have a job which they think exposes you to TB. I mean, that's the problem I have with veterinary students, is that one of the things that listed on there is vets. And my argument is actually vets are much more likely to get human TB than they are to get bovine TB, unless you're an idiot. So I didn't, and there are side effects associated with BCG. And when you get down to the kind of low levels of TB in people in most areas that we have, then the side effects are a bigger risk than the protective effects you get from the vaccine. It's a pretty crude vaccine. I mean, it's great because it's one of the first ever vaccines, but it's pretty crude. I can witter on if you really want to about Vol TB and BCG and all that kind of thing as well, because it's a fascinating history to it, BCG vaccination, and why we give it the way we do, because it works much better if you give it orally. But we don't.
Shall we move on?
And if you drop me an email, I'll have a more sensible discussion with you, if you like.
Okay, we'll move on to our next question, which is from Lauren. I will just try and find Lauren in the list. And Lauren, again, I'm allowing you. Oh no.
So while you're finding Lauren says, has anybody looked to see if the spread of TB has any relationship to cattle being moved between farms? And the answer is yes. So at the very beginning, I said that those sporadic outbreaks in cattle in otherwise low risk areas, so Lincolnshire, or they still have outbreaks in Scotland, they're not completely clear. people tend to look at the spoligiotypes from those cattle and say, oh, that's interesting. It's a grey one. It's spoligiotype 25. So that probably came from northwest England. And then they go into details of where that farmer's been buying his or her animals from. And usually that checks it out. More recently, there's been a policy of risk-based buying. So farmers shouldn't be moving animals from more dangerous areas to cleaner areas, because one of the problems has always been that, well, you might say, why would a farmer do that? And the answer is because cattle from, let's call them dirty areas, cattle from areas where there's a higher rate of TB would be cheaper than cattle from an area where there's a lower rate of TB, but your individual chance of importing TB onto your farm was relatively low, and there's compensation. So there's a kind of a free insurance policy in there. So they've tried to change that a bit, not least by insisting that farmers test their cattle before they're moved, and you're meant to test the cattle after they've arrived as well and keep them in quarantine and that kind of thing. And all of that long distance spread has been blamed on the long distance movement of cattle. I don't know if I'm talking out of turn or not, I don't care. Anyhow, so here at Nottingham, we have our own university dairy farm, and they do lots of work on welfare and reproduction and all that kind of thing. And occasionally we have an outbreak of TB, and it's nearly always a spoligiotype from somewhere else in the country, but we have a closed herd. We don't bring any cattle in, and haven't for years and years and years. So everybody gets very puzzled about that, and occasionally people say, oh, it's the bloody badgers, and I say, well, it isn't, because we've tested the badgers around here and they don't have TB, and if they did, it probably wouldn't be that strain anyhow. And that's another reason why I quite like that theory that I was putting out about transmission in soil and transmission in particularly in protozoa that can be moved around in soil and can last for an awful long time. That's answering even more questions than Lauren asked.
Okay, I'd just like to say, yeah, as I said there, the test pre-movement is only picking up 50% of the infected cattle. So infected cattle are moving, every week, every month around the UK. And then, that rightly there is, as I think, Lauren says, the post-movement test is using exactly the same test. Again, that's only going to pick up another 50%. So it's still going to be leaving behind overall around about 25% of infected cattle are being moved, being tested, and they're still there on the new farm. They'll cause a breakdown that will spread to badgers and a whole range of other wildlife. There's many, many species. A whole, hedgehogs, rats, deer, voles, shrews, all sorts of things that can get TB.
Our tyres. I'm going to come back from my environmental thing.
From the postman, from the milk van, all sorts of things. But yeah, moving cattle around, that's really, the veterinary profession in particular really has to get a grip on that and not permit that to carry on. It
always puzzles me, and I don't know what the cure to it is, because I don't really understand how it happened, why we move so many animals around, not just within the country, but internationally. How we export nearly all our sheep overseas, and yet nearly all the mutton we eat in Britain is imported. And how does that economically work? And the whole cattle industry seems to be based on moving cattle around, and you kind of think, why? How do we ever get ourselves in that kind of position? But it's a recipe for disaster if you have an infectious disease, any infectious disease, not just TB.
Okay. We'll move on to
the one after next. What is the vertical scale? On what? Karine, tell us.
I think that was perhaps relating
to
Ian's graph at the time. So
that was Ian's made up data.
Yeah. We'll move on to
the anonymous attendee,
Ian. Yeah. Oh,
okay.
So
the question is, both high-risk edge and LRA areas have been doing gamma radial testing, increased skin testing, and yet only the areas which have been culling badgers for over 4 years have seen a significant % decrease. Okay, so I'm just going to show you another graph. Hopefully I can share this. Okay, so this is from the badger cull zones. I know your brains are a bit fried, but this is the prevalence. So the vertical axis is what percentage of herds have actually got TB. Prevalence is the real driver of this. This is the percentage, not the attack rate, which is a good subtle indicator. This is the prevalence. This is the driver. So if we look at Gloucestershire, it was, it had a very high, it had a higher prevalence in 2011. It gradually came down, started killing badgers here. It goes up and down a little bit. And then this, at the end of the Downs paper, the peer-reviewed paper that people talk about was to the end of, to the middle of the year in 2017. The year after, up to 2018, and we've got that data already, and this is government data, it's not my data, it's not our data, my group's data, this is the government data, it's gone up, the incidence went up by 130%, and so the prevalence is now higher than it was before culling began. So that's 5 years of culling. If you're a Gloucestershire farmer, you spend all this time on culling, and it's gradually gone up. So it's not true that in the areas where we've been culling for more than 5 years, it's going down. It's actually going up. In Dorset, it's going up after 3 years. And as I said, if you look at this, and in Somerset in red, it's about the same. But these dotted lines are cows that haven't been exposed to the full impact of badger culling. They're in the area, but they haven't had the full impact. They have not been there for the full 4 or 5 years. And you can see in all three areas, these herds in existence do worse than the herds exposed fully to badger culling. That should be enough for the government to break the code and go, well, this doesn't work. Look at this. This is really showing this doesn't work. So that comment, I'm afraid, just isn't true.
Thanks. We can get back to your video. Oh, there we go. Oh, I don't know. Is it still screen share?
Okay.
Oh, no. There we go. Okay, so thanks for that. Right, we've got a lot of questions in here, so unfortunately we're not going to have time to work our way through them all, but we'll try and get through a couple more.
Shall I go for Roger's, the first bit of his question? Certainly.
Yeah.
Of the 8% positive, how many infected, how many infectious? And the answer is I don't know, and I don't think anybody knows. So on the one hand, you could argue that because these badgers largely had no lesions at all, or if they had any, it was microscopic lesions in individual lymph nodes, that there's no way those bacteria are going to get out into the environment and infect another badger. However, we only know where the lesions and the bacteria were where we tested. So if there were bacteria, for example, growing in the gut, and we didn't test the gut because it's full of other bacteria and it makes it really difficult to identify mycobacteria out of it, then that they would be shedding, and we just wouldn't know about that. And because I think we don't, and there may be other people out there who want to argue with me, but I don't think we understand enough about the pathogenesis of mycobacterial infections in general and TB in badgers. And I would say more generally, actually, we often know a lot about pathogenesis of disease and bugger all about how the bug escapes from the animal, human or otherwise, in order to infect other things. And the second one is not necessarily colostrum, but many TB, maybe most TB infections are latent for ages and ages and ages and ages. So I'm trying to remember what the figure is for human TB. Is it not something like 70% of us globally are infected with TB, but most of us will die of something else before that latent infection turns into an obvious disease.
Yeah, thank you very much for that comprehensive answer. I think we've got to talk again. How about the second half of my...
Oh.
If you mean the colostrum bit, I think the answer to that is simply that regardless of colostrum, most animals, including humans, that become infected with mycobacterium tuberculosis complex bacteria are latent for a very long time. And unless something else makes them more likely to develop clinical tuberculous disease, they will die of something else first. So TB is a big problem in poorer parts of the world, not just because more people there are infected, but because there's poor nutrition and HIV and a whole load of other reasons why they become clinically ill. Whereas in this country, the majority of people who get TB will never know they had TB.
Yeah, thank you. Yeah.
Okay. We'll move on to Alison's question. What is the main driver for the better bTB testing not being used extensively for cattle?
Okay, so that's a very complicated question. Obviously, there's a cost implication of doing the extra testing. But suppose you suddenly roll out really sensitive and really specific testing across the whole of England, you're going to find tens and tens of thousands of infected cattle. So if you kill all those cattle, it's going to be a bloodbath. It's going to break farmers' hearts. It's going to be incredibly destructive to do that all in one go, and it just isn't feasible to do that. So you might think that's a kind of mealy-mouthed answer or whatever, but it's actually true. Whichever side of the debate you're on, it's not possible to do that all in one go. But what you do want to do is start testing in an intelligent way that picks up the maximum number possible without breaking, the back of farmers. They've done all this badger killing, they've done all the testing, they've jumped through all the hoops. The government can turn up on their farm and kill their cows. So they're living on a knife edge. It's not surprising they're going out shooting badgers in the middle of the night. That's all the government's giving them to do. So it's a complicated question, but we've got a really, really accurate, specific, sensitive test in Activage. You could, for example, instead of doing all this silly Edwardian skin testing, you could use Activage on a bulk milk tank and find out if there are TB infected cattle on that farm using that screening test instead of this massive palaver with the skin test. And it's the 21st century. We can't pretend it's 1908 when the skin test was discovered. We can't pretend it's the Crimean War when this policy of stamping out, was initiated. You can't kill your way out of trouble. This is a modern veterinary profession. It's not acceptable just to kill animals, kill badgers, to kill cattle. That's not an answer. It's not getting us anywhere. We need to look at what's really causing this problem. And what's causing it's not just the bug, it's the fact that we're farming in an unacceptable way. We're trying to extract too much milk from dairy cattle. They're economically, they're being overheated, they're being overstressed. They can't produce that much milk. They don't get to bring their calves up. They don't have any sexual relations with the bull. They're living in hell, and that's why they're getting TB. Slaves got a lot of TB. It's not just the testing, it's the way we're farming, and that needs to change. Dairy needs to change, as simple as that.
Okay. Okay, we'll go for one more question, because otherwise we could be here.
Harry, I might have just deleted somebody's question by mistake. I was trying to type an answer to it, and it's gone.
Really
Sorry.
I've tried to take a copy of all the questions, so I can always email these to you and Ian later, and then perhaps you could write down some answers to them, and we could share them later. I've lost where we got to now, so we've just had Alison's question. Okay, so we'll have Debbie's question. Can a badger bTB vaccine pose any risk to a badger?
I think Malcolm's probably best to.
Oh, thank you. The answer is it shouldn't do. And you'll probably have seen that people are starting to trial badger vaccines in cattle now. So the issue with BCG, I talked about side effects before in humans, is mainly if those humans are immunosuppressed in some way, in which case you could, at least in theory, develop TB because it's a live vaccine. But it's knackered, which is why it doesn't grow very well in macrophages and so on, isn't able to cause disease. The issue with vaccinating cattle has always been distinguishing cattle which have been vaccinated from cattle which have been naturally infected, because, as Ian was saying, the diagnostic test is based on an immune response rather than detecting the bacteria itself, which, to be fair, is true also in humans on the whole. You would only isolate the bacteria from humans either at post-mortem or if they were ill enough to be coughing it up in spit. And the other issue with cattle vaccination is ensuring that the BCG isn't in a bit of the cow, which then goes to slaughter. So you have to make sure that it can't get into the food chain in case somebody who's immunosuppressed then eats that bit of cow.
Lovely.
More answers than questions.
But just, you know, there's another question later on about cattle vaccination. Cattle vaccination has been 5 years away. I was talking about this with Malcolm. It's been 5 years away since I was a vet student. There needs to be a political will to vaccinate cattle, and all the evidence suggests that that is going to cause a decrease in incidence of between 60 and 70%. Where it's been trialed in Mexico, for example, that's the findings. So we should be rolling that out as soon as possible. The reason it's not being rolled out so far is because it interferes with the silly Edwardian skin lump test, because that's measuring an immune response. If you move to a proper DIVA test, like a differentiating infected from vaccinated animals, you can roll out BCG more or less straight away. But it's still five years away. There needs to be a political will to do it and to protect cattle.
Well, I'm looking shifty because I'm trying to look at my phone and see what APHA have tweeted recently, and I don't want to talk about things that it's not 5 years away. There's a DIVA test and there's a vaccine, and they're about to be trialed. But I'd rather that bit didn't go out on YouTube because I shouldn't have said it. Whether it'll work or not is another matter, but yeah.
Okay, brilliant. I think we'll start wrapping it up there, unless you can spot any questions that you could answer very quickly.
Okay, there's a couple there. There's a couple there from Jay Bradley. Oh, he's actually to Malcolm. Yeah, the reason they're being culled is because it's an extermination policy. It's got nothing to do with the fact. What they're trying to do is manufacture consent for this culling policy. It's not to do with the fact that badgers are giving these to cattle. It's if the cattle are there, they've got TB, they give it to badgers. Even though they know that that's been caused by cattle movements, they're still going to cull badgers because it's an extermination policy. It's not to do with disease control. It's politically motivated. So that's the simple answer to that. It's very sad, but that's true.
My simple answer is I don't know.
Can you hear me now, Malcolm?
Yes, hello.
Hi, Malcolm, it's Janice. Yeah, the reason I wanted to flag that up is because we have repeatedly requested evidence from DEFRA, the APHA and Natural England about what evidence they have used to justify culling in the edge area, including in our Notts and Leicestershire vaccination area. And they've repeatedly told us that they can't release evidence to us, or in fact, they haven't completed the epi maps yet, the epidemiology maps showing claimed endemism in badgers. So we're at a complete loss as to what evidence they use. So clearly I'm moving towards Ian's position that it's a political decision, not a scientific one, because they can't produce any evidence for us. So they've now had something like 7 or 8 freedom of information requests for evidence as well.
Oh yes, I get lots of those as well. So sorry to those of you who I've not not released everything as raw data. Well, I'm not really that sorry. Just anyhow, so there have been some DEFRA funded APHA carried out roadkill badger studies recently, and I think is there not one being maybe not peer review published, but a report that's been published about finding some badgers that were positive in Lincolnshire. But that's a really, really specific answer. I suppose I think the question you might like to ask is, what is the evidence for saying that the badgers have TB? Not what is the evidence for the badgers having TB and waiting for those maps, but asking what makes a badger positive on those maps? What you know, is that because somebody's isolated TB from a badger, or what?
Okay, thank you, Malcolm.
Okay, well, thank you, everybody. So we'll leave it there. I have quickly tried to copy everyone's questions into a document, so I will pass these on to Malcolm and Ian, and we will try and post some answers onto our website, or get them all out to everybody in an email who joined the talk this evening. So thank you everyone who joined us. We had well over 100 people at one point. A few people have dropped off into the questions section. But thank you to Malcolm and Ian for joining us this evening. Really informative talk, and there's lots of great comments coming in at the end there just to say how much people have enjoyed it and how informative it was. So I'm sure everyone's taken something away from that. David, do you want to just wrap up the evening for us?
Yes, I will, very briefly. It's a shame we couldn't have more questions. There are a lot to be asked. I had a few myself, but it's been fabulous to have Malcolm in particular, and also the bonus of Ian along tonight. Been waiting to see Malcolm's final talk, or final talk, for a long time. And the one question I'm going to leave open, I'm not looking for an answer. I'd have asked Malcolm, is it right to be culling badgers in Leicestershire and the other edge counties, apart from possibly Cheshire? But I won't get an answer from you tonight, and you probably wouldn't give one, but it's obviously a very complex scientific situation. We're seeing it the same with COVID. There's political and scientific interpretations. But thank you for a very, very good evening, very interesting, and we look forward to some more in the future. Thank you both.
Thank you, everybody, and enjoy the rest of your evenings.
Thank you, Harriet.
Thank you very much. Thank you, Malcolm. Thank you, Ian.
Thank you.
Bye.